Hello and welcome to Bio40 Pre-Lecture 32. Time to see what happens when the last two Hardy-Weinberg assumptions fail to hold. Previously, we've covered the effects of selection, mutations, and small populations. Today, we're going to start with migration. Like the previous violations of Hardy-Weinberg assumptions, migration is capable of changing allele frequencies. A new group joining a population can alter allele frequencies, and is also a way that new alleles can be introduced into the population. A small group migrating away from a larger population and becoming isolated can also have interesting effects. Take this population that is living on an island. For a certain gene, there are three alleles: a blue allele, a yellow allele, and a red allele. Now imagine that a small group decides to head out and settle a new island. We call them a founder population. The founder population won't necessarily be representative of the population they separated from. In this example, it happened to be that none of the individuals with the yellow allele settled the new island. That allele is completely non-existent in the new population. However, an individual with a rare red allele did happen to be part of the founder population. Because the founder population is small, it will be subject to genetic drift. So it's possible that the red allele will quickly become extinct, but it's also possible that its frequency could greatly increase. The selective pressures of this new island could also favor the spread of this previously rare allele. The allele frequencies of the population on the new island are now very different from those of the original population. But then suppose another group from the original population migrated to the same island. This would alter the allele frequency of the second population. It's also a way that the yellow allele could be introduced to this population. Alternatively, imagine a scenario where the individuals on the new island are unable to make contact with the original population for millions of years. We've been looking at allele frequencies of this one gene, but the same thing could be occurring in thousands of different genes. Different mutations will appear in the two populations. It's likely that the islands will have different selective pressures. Maybe there's a temperature difference, so one favors individuals adapted for warmer climates, and the other favors those who can deal with cooler conditions. Maybe there are different food sources that favor different genotypes. Over vast stretches of time, the populations will become more and more different from each other, until they eventually become incapable of mating with each other. At this point, the two populations are different species. Reproductive isolation is key for speciation, the process of a single species becoming multiple distinct species. The other processes we've talked about can cause a species to greatly change form over time, but as long as all the individuals within the population interbreed, it will remain a single species. Only through isolation, usually the result of a geographical barrier, can a population split into different species. This is what Darwin saw with the famous finches on the Galapagos Islands. Populations of finches on different islands face different selective pressures, such as the food sources. They develop different traits to best adapt to their specific environments. This was an important observation toward the theory of evolution by natural selection. So this just leaves one more topic to address: non-random mating. Specifically, we're going to address bias toward mating with distant relatives versus bias toward mating with close relatives. The former is called outbreeding, and the latter is called inbreeding. Note that all matings occur between relatives because all individuals within a species are related. In fact, all life on Earth is distantly related, but there are problems associated with mating with close relatives. Recall Mendel's crosses from the very start of the course. When Mendel crossed two different true breeding lines, the offspring were all heterozygous. Outbreeding tends toward heterozygosity, but in a self-cross, heterozygosity decreases. After one generation, half the offspring are homozygous, and the fraction of heterozygotes decreases by half with each generation of self-crossing. Self-crosses are the extreme example of inbreeding, but inbreeding in general reduces heterozygosity. So non-random mating alters genotype frequencies from what would be predicted by Hardy-Weinberg. Outbreeding leads to more heterozygotes than would be predicted, while inbreeding creates an excess of homozygotes. This alone does not change allele frequencies. However, this can happen when selection is also involved. Selection against heterozygotes is uncommon, but we've discussed many scenarios where homozygous recessive genotypes can be detrimental and even lethal. Let's go over some inbreeding terminology. Matings between close relatives are called consanguineous, and the offspring of such matings are called inbred. Such offspring are more likely to be homozygous because they can inherit the same allele from each parent. Take this simple pedigree. For a certain gene A, let's say the father in the first generation has alleles A1 and A2, while the mother has alleles A3 and A4. If their children mate, what are the odds a grandchild is homozygous for allele A1? Well, there's a 1/2 chance this daughter inherits an A1 allele from her father. Likewise, there's a 1/2 chance this son gets the A1 allele. If both those events occur, there's a 50% chance each parent passes the A1 allele to the grandchild. So the overall odds are 1/2 times 1/2 times 1/2 times 1/2, or 1/16. There are the same odds that the grandchild is homozygous for A2 or A3 or A4. So overall, there's a 1/4 chance they'll be homozygous for some allele. This number, the probability that both alleles in an individual are identical due to descent, is called the coefficient of inbreeding. It's given the symbol F. We just saw why the coefficient of inbreeding for sibling matings is 1/4, but there's a different value for each type of consanguineous mating. Note how the value decreases for more distant relatives. We can use the coefficient of inbreeding to calculate the frequency of homozygosity. The frequency of homozygous recessive individuals is QF plus Q squared times 1 minus F, where Q still refers to the recessive allele frequency. For non-inbred individuals, F is essentially 0, so this equation reduces to Q squared, the same as you'd expect from Hardy-Weinberg. But for inbred individuals, the first term of the equation is much larger than the second because Q is much greater than Q squared. So the whole equation reduces to approximately QF. By taking the frequency of homozygosity for inbred individuals and dividing by Q squared, the Hardy-Weinberg frequency of homozygosity, you can see how much more likely an inbred individual is to be homozygous compared to a non-inbred individual. Let's look at an example. Albinism is a recessive condition with an allele frequency of 0.01. What are the odds that the offspring of two cousins is albino? We can calculate the frequency of homozygous recessive individuals with this equation. We can work out the coefficient of inbreeding just as we did before, or we can just consult the table from a couple slides ago and see the value for cousins mating is 1/16. And the problem tells us that Q is 0.01. So plug in those numbers, and we get 0.00072. Compare that to Q squared equals 0.0001 for non-inbred offspring. The children of cousins are 7.2 times more likely to have the condition than the children of a random mating. Let's observe how this ratio changes as we alter our parameters. The more distant the relation is, and thus the lower the coefficient of inbreeding gets, the smaller the ratio becomes. Relationships more distant than second cousins often do not have too large an effect. The other factor is the allele frequency. The ratio increases as Q decreases. When Q is large, the allele is common in the general population. There is already a fairly high chance of having homozygous offspring from random mating, so inbreeding does not raise the probability too much. But the smaller Q is, the less likely it will be to get a homozygous recessive child from random mating, so the effects of inbreeding will be greater. Those are the two major relationships to be aware of. The relative probability of being homozygous recessive decreases when the coefficient of inbreeding decreases, but it increases when the recessive allele frequency decreases. This brings us to another Bio40 life lesson. Inbreeding. Maybe don't? So that's the last of the Hardy-Weinberg assumptions. We've covered genetic drift, how alleles can be lost or fixed just by chance, a consequence of populations not being infinitely large. We just finished looking at how non-random mating alters genotype frequencies. specifically how inbreeding creates excess homozygotes. This alone will not affect allele frequencies, but some homozygotes are subject to a selective disadvantage. We talked about how mutations are the ultimate source of new alleles, but how this has a small effect on allele frequency unless combined with selection. We saw how migration can introduce new alleles to a population, while reproductive isolation can lead to speciation. And finally, we addressed selection, how environmental conditions favor some genotypes to survive and reproduce over others. This is the force that drives adaptive evolutionary change. That does it for population genetics and our penultimate lecture. Just one more to go, the class-selected topic of designer babies.